Hello everyone, welcome to our channel Love of Biology. So today we are going to learn about basics of genetics. What is genetics? It is nothing but a study of inheritance and variations from one generation to the other. So father of genetics is Gregor John Mendel. The term genetics was given by Bateson and he is the father of modern genetics. So father of experimental genetics is Morgan, who worked on Drosophila, a fruit fly. So experimental proof was given by later by the Morgan. So next, so Mendel, he chose garden pea plant for his experiment to study the inheritance, right? So based on few characters, those are, it is an annual plant, so he can observe its life cycle in a few months. It will undergo self-pollination naturally. That means it can undergo self-pollination and we can perform the cross-pollination also. It is easy to cultivate and it produces large number of the seeds within a short time. It has a many contrasting traits, that is 7 contrasting traits it contains. Okay, so let's see what are those 7 contrasting characters. So first one is seed shape, round is dominant over the recessive wrinkled, and seed color yellow is dominant over the green recessive, and flower color is dominant, that is flower color purple is dominant over the white recessive one, and pod shape inflattened is dominant over the recessive constricted one, and pod color, that is yellow, is dominant over the green color. So these characters, each character will have the 2 contrasting characters, so dominant as well as recessive. Our position axial is dominant over the terminal, and stem length, that is tall, is dominant over the dwarf. So these are the 7 contrasting characters which are present in the green plant. So on these characters Mendel worked, he observed for the inheritance patterns. So let us see his technique. First he choose a parent plant which are homozygous characters. He will choose so he cross-pollinated them by using the artificial hybridization to get the F1 hybrids. So after F1 hybrids production, so he self-pollinated these F1 hybrids and he got the F2 hybrids. So among these F2 hybrids, by using these ratios, he observed the inheritance pattern. So this is the Mendel experimental technique. First he used cross-pollinated, later he self-pollinated, he observed for the results. So let us see few new terms. What are these homozygous or heterozygous characters? Let us see. So gene, what is gene? First of all, gene is nothing but a small segment of the DNA. So gene contains 2 alleles. 2 alleles are alternative forms. So homozygous condition, if these 2 alleles which are present in the gene are similar, okay, then we call it as a homozygous condition. If they are dissimilar, we call it as a heterozygous condition. So these alleles, there are 2 types, dominant as well as recessive. Dominant alleles means it can express its character in both homozygous as well as heterozygous conditions. So one allele is enough to express its character, whereas the recessive allele, it cannot express its character in heterozygous condition. It can express only in homozygous condition, that is it can't express its character if one allele is present. Both should be present, both homozygous ones should be present, then only it will express. So thank you for watching. Please subscribe my channel. Thank you everyone.